In 1996, a communion wafer in Buenos Aires turned red. It's said that scientific testing proved that it was living human heart tissue. But is that the full story? There are already a lot of videos on this subject, but I've found that they leave a lot of information out. I'm going to try and give you the full picture, complete with some home experiments to explain what testing was done and what it found. While there is a lot of information on these events online, much of it appears contradictory. To cut through the contradictions, I am only considering references where the source of their information can be verified. This leaves me with these main sources. The first is information from the church itself in the form of a website and a YouTube channel. Only locals or people with a VPN set to Argentina have access to the website. This is supposedly because the church has had a problem with satanic hackers. You know, the kind that doesn't know how to use a VPN. Next is a group that investigated the miracle claims in 1999. They interviewed witnesses and collected samples for scientific testing. 3 members of this group have produced books and videos on this topic. The first is Dr. Ricardo Castañón Gómez, who has a PhD in clinical psychology. He has written several books and has recorded many interviews in Spanish. Ron Tesorero is a lawyer from Australia. He has written 3 books on this subject in English, plus several videos. Mike Willesee, a journalist from Australia, accompanied Tesorero. He has produced documentaries on the subject, and it is also discussed in his autobiographies. The other popular sources are more distant from the events. Dr. Franco Serafini, a cardiologist, wrote a popular book that was published in 2021. While he did visit the site himself, it was decades after the event. His 2 main sources of information are the work of Dr. Castañón and Maurizio Blondet, an Italian journalist that seems to be best known for anti-Semitic 9/11 and UFO conspiracy theories, which makes it a bit concerning that Serafini describes him as one of the best Italian journalists. I decided that Blondet's book was not worth my time. His investigation began in 2013, 17 years after the 1996 incident, so I doubt he has much reliable information that's not presented in earlier sources anyway. This has made me hesitant to use Serafini because it's often unclear where he is getting his information from. I'll let you know when that is the case. Another source that many of you may have seen is the Eucharistic Miracles of the World posters and website originally produced by the soon-to-be canonized Carlos Acutis. The information on this website has changed a lot over the years, presumably to correct inaccuracies. At this time, they no longer contain any information that is not already in one of the previously mentioned sources, so I won't be citing this source as it is redundant. With that being said, let's move on to the miracles. Our story begins on May 1st, 1992, at the Santa Maria Church in Buenos Aires. To those of you about to comment, the Eucharistic miracle of Buenos Aires was in 1996, not 1992. That's because the one that you've heard of is the 5th claimed Eucharistic miracle at this church. This fact leads to a lot of questions. Why have there been 5 claimed miracles here? Is this God's favorite church? Is an individual at this church committing fraud? Do the members of this church just have a tendency to see miracles in everything? Speculate on the answers yourself. But I do think that this context is important, and it's interesting that so many sources only mention the 5th incident. After all, it seems fairly unlikely that the first 4 miracles weren't authentic, but the 5th one is, right? Does God have a punch card, fake 4 miracles, and get a real one free? I've heard that God works in mysterious ways and all, but this seems unlikely. The incident began when a priest found 2 pieces of communion wafer on an altar cloth after communion. This image is pulled from a video of a presentation at the church. I'm not sure why it exists. Back in 1992, you didn't just pull your camera phone out of your pocket and snap a picture. Cameras were their own thing, phones were attached to walls, and film was expensive. People weren't just snapping pictures of their food all the time. While an apparent miracle would certainly be photo-worthy, the pre-miracle wafer pieces would not be. This seems so odd that I have to wonder if the image is from some kind of reenactment, although it's not presented that way. The wafer pieces were placed in water to dissolve and locked in the tabernacle, which they say is protocol for the Catholic Church. This is mostly true. Consecrated wafers are supposed to be eaten whenever possible. Referring to the Roman Missal, placing the wafer in water is the procedure if the wafer becomes poisoned. Not necessarily literally, but it is generally agreed that this procedure is for when it is not possible to eat the wafer without risk to your health. A common example given is when a wafer falls out of someone's mouth onto a really dirty floor. In this case, the wafer was on an apparently clean altar cloth. It is unclear why the priest didn't just eat the wafer. The second part of the procedure in the Roman Missal that there seems to be some debate over is how long to leave it sitting in water. Of the churches I could find protocols for online, many say that the water and wafer can be dumped down a special drain called a sacrarium after an hour. Others say to break it up or stir it to make it dissolve faster. So I'm unclear how prevalent the practice of just letting a wafer sit in water for weeks on end is. Certainly, stories like this pop up often enough that it must not be unheard of, but it's far from true that this is a universally followed protocol. When the priest checked on the wafer a week later, he found that it had not yet dissolved. Many videos suggest that this is unusual, which is weird because I think most people know that bread does not dissolve in water. What may be causing some confusion is the fact that communion wafers can dissolve in your mouth. Your saliva contains an enzyme called amylase, which breaks down the insoluble starch into sugars which do dissolve. So in most cases where the church is trying to dissolve a wafer that has fallen out of someone's mouth or that an elderly person was unable to swallow, those wafers would dissolve. It is also possible that a wafer left sitting in water for a week could dissolve due to microorganism growth, as some microorganisms produce amylase. Attempting to repeat this myself, I've obtained unconsecrated wafers and placed many in water to see what happens. For years I had never seen an unlicked wafer dissolve in my experiments, some of which were left sitting intact for months. However, more recently, every wafer would end up dissolving within a week. Presumably, this is related to a change in the microorganisms growing on the wafers. Instead of a dissolved wafer, the priest noticed what appeared to be 3 blood clots covered in a white fuzz. And we can see that these pictures look pretty impressive. I can understand why they found a camera in 1992 for this. But does impressive mean supernatural? A lot of people telling this story seem to think so. Tesoriero says, "It goes without saying that the properties of bread are not such that when placed in water for a week, one would expect a bright red substance to come from it." But I don't think he has much experience with wet bread. To examine the possibility myself, I've put over 100 unconsecrated communion wafers in bowls of water over the past 3 years. Of those, about a dozen have turned red, including the very first wafer, which I carefully tipped out of the sealed packet into a sterilized container filled with tap water. I was not alone in this investigation, as a recent study performed a similar experiment. My success rate was only about 1 in 8 wafers turning red, slightly lower than they reported. But I also didn't leave the wafers in dirty locations, and my room temperatures were lower, which is not ideal for bacterial growth. Most of them only grow small red spots before they dry out. However, under the right conditions, more impressive specimens are certainly possible. For the wafers in these time lapses, I subcultured the microorganisms from one of the wafers that had previously turned red on some agar. Then I touched the colonies on the agar to a wafer before placing it in water. This practically guarantees that the wafer will turn red, which makes it practical to do things like week-long time lapses and filming videos of experiments. I will put N's on the completely natural images and S on the ones I gave a kickstart to so you can tell the difference. You'll notice a lot of fruit flies are joining in on this experiment. They are attracted by the yeasts growing on the rotting bread. Because these did originate from a wafer that just turned red on my counter naturally, I can't be certain what is growing on them. One species of bacteria called Serratia marcescens is particularly known for turning bread red. But I have a few reasons to think that this is actually a fungus called Epicoccum nigrum. I'll discuss why I think that in a future video. The sample I've grown that looked the most like Buenos 1992 is this one. On this wafer, the red was growing inward from the outside with a bit of fuzzy white fungi. Then, the inside of the wafer dissolved, presumably because a different microorganism started producing amylase. This resulted in floating pieces that were all red, with a fuzzy appearance at the edges. Unfortunately, it was winter, and I was growing the sample on the floor closer to a heat source, and let's just say robotic vacuums don't respect replica miracles. Thus, I lost the chance for photos or video with this particular sample. Comparing these pictures to the pictures of the 1992 specimen, I think you can see that this looks pretty similar, right down to the white fuzz. So this seems like a pretty likely natural explanation so far. But let's get into the scientific tests. The 1992 Buenos Aires wafer was then tested for blood using a reagent strip by Dr. Botto, an oncologist from the congregation. I haven't found any source that gives the details of this test, but it sounds like she used test strips she happened to have handy. This most likely means Comber test strips, which are used to test for blood in urine. I've used these tests on my wafers, and blood is this middle one, which will turn green within 30 seconds if it is positive. So you can see the first one was clearly positive. The rest stayed pretty yellow, but when I held them up to the comparison chart on the bottle, they were actually the color for trace amounts of blood, with the bottom one having slightly more. Most of the samples I have tested have been at least slightly positive. So what's going on? Comber test strips are very sensitive. They will change color if even a tiny amount of blood is present. Although this has been sitting uncovered in my apartment for a week, I am quite confident that it has not been contaminated with blood. So I don't think that's the problem. But Comber test strips aren't very specific, which means there are many other substances that will give false positives. These can include a number of common household substances, including cleaning agents like bleach. So it's possible that the positive is coming from traces of something left in the plastic bowl or on the wafer itself. But I think the culprit here is probably one they warn about right on the package, microbial peroxidases. These are substances produced by microbes to aid in digestion. And we obviously have a lot of microbes on these wafers. To test this, I am going to test the same wafer twice, once immediately after being put in the water and again a week later. You can see that the first test gave a negative result, so now we know there is nothing on the wafer, container, or tap water that is giving the positive. But a week later, there is a strong positive. So even though this particular wafer did not turn red, clearly microorganisms give a false positive on the Comber test. So far we haven't seen anything in the 1992 tests that would rule out an ordinary rotten wafer. What's next? Serafini says Dr. Botto then tried to draw up some of the red stuff in a syringe, but couldn't do so because of its hardened consistency. Note that this is one of the details I could not confirm with any other source, so it's not clear where Serafini was getting his information. I also don't know if there was a needle on the syringe or what bore size she used. Trying on my own wafers, I can draw up this looser cloudy red material, but not the more solid main part of the wafer. Serafini then says she even claimed she had seen muscle cells and living fibrous tissue under her microscope. There are no details provided regarding her methodology, and no other sources mention it. Other experts will examine the specimen under the microscope and provide more details later. We will look at my samples under the microscope then. On May 10th, 1992, a week and a half after the wafer was first put into water, a red substance was found on the ciborium and paten, containers that hold consecrated wafers. These were incidents B and C. I admit that counting these as separate incidents is a bit of a stretch, but Serafini counts them separately, and for clarity I would like to have a separate letter for each specimen. Next, the church asked a hematologist named Dr. Sassett to examine the samples in her lab. Castagnon only includes 3 pages of her 92-page report, so there is a lot of context missing. It's also written in Spanish, which causes me difficulty to begin with, but I had particular difficulty interpreting this document. Even with the help of a native Spanish-speaking friend, I can't promise that I've interpreted everything correctly. Of the 3 pages we have access to, the first page is dated June 1992. It reads, Preparation number 1, the same was extracted from one of the patens 3 days after the appearance of a bloodstain on it. This seems like an odd way to start a report, but she may have been referring back to a previous page of the report that Castagnon doesn't include. She then gives a leukocyte formula, which is essentially a white blood cell count, and notes that it's human blood contaminated with debris and bacteria. She does not say how she concluded it was human. Castagnon says this leukocyte formula is for the 1992 communion wafer, which I'm referring to as Incident A. But this report clearly states that the sample was taken from a paten, and Incident A never had anything to do with a paten. The wafer was found on an altar cloth and then placed in a clay bowl. Therefore, I am quite certain that this page is referring to Incident C, the blood stain on the patent. Castagnon never mentions incidents B and C at all. Perhaps he didn't know about them and was confused. Alternatively, he could be trying to keep incidents B and C secret for some reason, which might explain why this part up here says our classified sample. But then why would he include this page at all? The image on the top left is of the 1992 sample after it dried, but this report was written before the sample dried out, and I think everything above this line was added later by Castagnon. The second page is about the material that was in the baked clay container. That description matches incident A, the communion wafer. She starts with macroscopic observations, which basically just means that she looked at the sample with her bare eyes. We have a picture here so we can look at it ourselves. She says it looked like a clot. Here is a blood clot from a chicken heart for comparison, and here is my moldy wafer. She then says it also had the consistency of a clot when touched. I did squish the slime. I'm going to put that footage in a separate video for ASMR fans and masochists because it's pretty disturbing. The chicken heart felt like meat, I'm sure you're familiar with that. The blood clot was more rubbery than I expected, but it had been frozen and I'm not sure if it's really a useful comparison of a blood clot beyond its color. The wafer was very soft, more like a fresh blood clot. Next, she says in one area there was a white formation that resembled the branches of a bush. We can see that pretty clearly on this sample. It's fungus. Obviously, there's none of that on the blood clot or the heart, although I suppose there might be if I let them decompose. She says in other places there was a blood red color. That's obviously in the blood clot and the rotting wafer, but the heart tissue is muscle, so it's more of a duller pink-purple color. The next sentence Serafini translates as the substance remained fairly stable until holy water was added to it. Let's try that and see what happens. Obviously, that was a joke. In reality, nothing happened. Of course, nothing actually happened to the real 1992 wafer either. What this sentence actually says is that the wafer remained more or less the same as long as she kept adding the holy water, but when she stopped, the sample started to dry out. That's much less exciting. Dried heart tissue is basically jerky, dried blood clots become hard, and the rotting communion wafers cling to the container as they dry, then eventually start to peel off. That is exactly what the Buenos Aires sample did. Then she looks at it under a microscope with 16 times magnification. This sounds simpler than it is. When you think of a microscope, you probably think of a transmission light microscope like this one. And I also initially assumed that's what she meant. The light source is at the bottom, and it is transmitted up through the sample, which allows you to see the details of the cells that make up the tissue. So if I just take some little chunks of each sample and put them under the microscope like this, you can't see anything, because the sample is not transparent, so the light can't get through it. You can see a little bit at the edges of the wafer here, because that is soft enough to form a thin layer. But in order to view this heart tissue, I would need to first process the tissue. First, I would add a fixing agent, such as formaldehyde, to freeze the appearance of the tissue as is, so that everything I do afterwards doesn't destroy it. Then I would dehydrate it in alcohol. Then I would use a clearing agent such as xylene, which makes the tissue transparent and allows wax to penetrate it. Then I would embed it in paraffin wax to make it solid enough to slice a layer a single cell thick under a microtome, which I could then mount on a slide, and then de-wax and stain. She doesn't mention doing any kind of processing. If her sample was a lot less solid than heart tissue, then she could have just squashed the sample under a cover slip to get something transparent enough to look at. That works with the sample from my wafer, but the heart tissue and even the blood clot are just too solid. This lens is 10 times magnification and the camera mount is another 1.5 times, which gives 15 times total. That's as close as I can get to 16 times. It's not much magnification. Generally, with a transmission light microscope, you are trying to look at the cells that make up the sample, and this magnification is much too low for that. She says the red formation had several planes. I'm not quite sure what that means. She's likely referencing focal planes, but all that would mean is that the red formation was more than 1 cell thick. She says the white branching structure seemed to be fungi or remains. That obviously matches what we're seeing because this is actually fungi. But it's not white because you can't see white on a transmission light microscope. White objects are opaque, which means you can only see their shadows. That, combined with the low magnification and the lack of notes on sample preparation, makes me think that she's actually using a dissecting or stereo microscope. These have 2 lenses, which allow you to see the object in 3D, and 16 times magnification would be more typical for this type of microscope. I don't own a dissecting microscope, but I do have a digital microscope I can use to magnify the sample, which will give you a similar perspective without the 3D. Unfortunately, it's a cheap one, so I can't tell you exactly what magnification this is, but the minimum is 50 times, so it's certainly more than the 15 times she was using. With this, you can clearly see the white branching fungi. I'm still not quite sure what she means about the multiple planes on the red formation. Then it gets really weird. I'm going to directly quote the Google Translate of this section. On one of the occasions when I was able to observe it, there was an area that seemed to pulse rhythmically. I haven't found anyone with experience doing benchwork in a lab that thinks this sample was actually pulsing. In general, the nature of microscopy work has you bent over with your eye pressed against an eyepiece, staring into a bright light through lenses. This is very unnatural and can cause you to see odd things. She may have been seeing the reflection of blood vessels in her own retina, or she may have induced a migraine with an aura, which would explain why a couple sentences later she says that she was feeling unwell and had to stop. So it's likely this observation didn't even correspond to anything physically pulsing. But if it did, could there be a natural explanation? Slime molds do pulse, but slowly, and the typical period is over a minute. She doesn't say how fast it was pulsing, and I suppose any movement might stand out, but it seems unlikely. A more likely explanation is all of those insects you've seen my sample attracting, their larvae, and their microscopic cousins. Their hatching, swimming, or even eating could appear like pulsing if they were inside or under the tissue. As far as heart tissue goes, fetal heart cells do pulse spontaneously. Adult heart cells don't. Don't ask me what miraculous Jesus cells would do. Next, she says, when this formation dried, it was seen how it was coming off in layers of a dark red color, and on them you could see formations as if they were threads of a lighter color, sometimes transparent, that were reminiscent of how you have seen mitochondria in photos in books. In other areas, I observed some formations that reminded me of drops of water, of which I saw at least 3. Mitochondria are much, much too small to be seen at 16 times magnification. The squiggles like she drew on this little sketch can't even be seen with a light microscope. So that's obviously not really what she was seeing. You can see these weird threads with the microscope in my dried sample. I assume those are just tiny threads shed off my clothing. Most of them are on the wafers, but there is one on the chicken heart. Perhaps she meant something like that. I'm not sure what she could mean about the drops of water. I see nothing like that on the dried wafer, blood clot, or heart. She ends the report by saying she had to stop due to health problems, as I mentioned earlier. Somebody should tell all the people who think this wafer has magic healing powers that. Overall, I would say her observations are more consistent with a moldy wafer than with either a blood clot or heart tissue, although there are certainly some unanswered questions. 2 years later, there was another claimed miracle at the church. On July 24, 1994, a priest reported seeing another drop of blood running on another ciborium, and this is incident D. In October of 1995, there was another report written by Dr. Sassett. Castagnon says that this one was 95 pages long, and his appendix contains 2 of them. Page 1 begins, presumably talking about her study in 1992. I was called upon to carry out studies in my field of expertise on a material whose origin I did not know at the time. I was notified after the first investigations had been carried out. This is an extremely interesting claim, because remember that part about pouring holy water into it? That's not exactly normal laboratory procedure for an unknown sample. So she clearly knew that the sample came from the church. She also presumably knew that they thought it was blood, because why else would they call a hematologist? So what exactly were they trying to keep secret? She then says that she did a blood smear of sample number 1. But we have no way of knowing what sample number 1 is. Is it the same as what she called preparation number 1 on the previous report, which was our incident C, the blood droplet from the patents? If so, why do it again? If not, there are now 3 other potential samples she could be referring to: the communion wafer, the 1992 ciborium, and the 1994 ciborium, and I have no way of knowing which it is she's analyzing. Castagnon again seems to believe this is for the communion wafer, but he was wrong for the first page, so I don't trust him. I'm also not sure how she could have done a blood smear on the communion wafer. To do a blood smear, you smear the liquid blood on the slide. But remember how Dr. Botto said she couldn't draw the sample up in a syringe? And Dr. Sassett noted that the substance had the consistency of a blood clot. And we know that substance doesn't dissolve in water. So how would you smear it? The numbers are fairly similar to the first blood smear. She again notes that the sample is contaminated with fungi and bacteria. Lastly, we have this page, which starts with, "A small portion of the original sample was given to me for further testing." This is again confusing because we have no way of knowing which sample was the original sample. It says the sample was placed in an unsterilized tube with holy water, and part of it was kept at room temperature for over a year. Later, when you're laughing at my improvised home experiments, I want you to remember that this actual doctor in an actual lab tossed a random sample from an unknown source in an unsterilized tube of holy water and kept it at room temperature for over a year. It then says, when it was examined again, it was found to have a faint pinkish color. The 4 samples that we know of are a communion wafer and 3 droplets of blood. So is she talking about a bit of the communion wafer, which started bright red, turning a faint pinkish color? That seems odd because the remainder of the wafer dried out and is still on display at the church, and it is still quite red. Or was a blood sample put into water and the water turned a pinkish color? But that seems too obvious to even mention. Or perhaps she's talking about some unknown 5th sample. Whatever this sample was, it tested positive for sodium and potassium, which would be true for either a communion wafer or blood, so that really doesn't tell us much. It had appreciable quantities of lactate dehydrogenase, but that's produced by all living cells, be they human or microbial, so that's not going to tell us anything useful either. Then an electrophoretic run was performed to demonstrate the presence of proteins. She refers to an attached study, which Castagnon doesn't attach. Gel electrophoresis is basically just a sorting method. Substances, usually protein or DNA, will move different distances along the gel based on their electrical charge and their size. Normally, this test is done on serum from blood, and because we know exactly what substances are in blood and how far each type moves across the gel, we can determine the concentration of each type of protein based on the intensity of the corresponding band. But of the 4 possible samples, we have 3 dried blood droplets and a solid red communion wafer. Serum is the liquid part of blood. It evaporates when blood dries, and it's the part that is left over as a liquid when blood clots. So none of these samples would have had serum. So either she's testing some unknown 5th sample, or she's just testing the holy water from the unsterilized tube as though it was serum. Obviously, that means that the concentrations are going to be all wrong, and that's presumably why she doesn't report any concentrations in the results. She says it was confirmed that the sample contained most of the proteins, except for traces of gamma globulins as observed in immunosuppressed patients. That would suggest that she got the first 5 bands, but this last smear was missing, which for a standard test would mean that the patient was likely immunocompromised. But the gamma band is just for the largest proteins. For blood left sitting in holy water for a year, it could just mean that those large proteins have degraded and broken down and thus are no longer big enough to show up in the gamma band. And if she tested something that wasn't blood in the first place, like my moldy crackers, anything biological in origin has proteins. My wafers are made up of wheat flour, which contains proteins including gluten. The bacteria and fungi growing on it also produce proteins. Without knowing the details of the test performed, I can't predict exactly what results you would get, but I guarantee you that you would get bands, and that many of them would overlap with the ones found in human blood. So if this test was in fact on the wafer, it would just be garbage in, garbage out. She ends by mentioning plans for future tests of DNA and blood type. I have no record of these ever happening. This is the last we'll see of samples B, C, and D. I do not know what happened to them after this. Of course, I think most people assume that those were just an altar boy with a paper cut, so I don't think anyone really cares what happens to them. Incident A, the 1992 communion wafer, was eventually allowed to dry out, and it went for testing at a forensics laboratory along with the sample from the fifth incident, the famous 1996 communion wafer. Did they finally do some tests there that could distinguish between my home wafers and human heart tissue? We'll find out in part 2. Goodbye for now.